We'll get started. Usually what I do is I start with a couple of announcements if I have them. So one is we're going to try the iClicker today. The reason I always wanted to, since I like the technology, and so I was going to update to the newest version of iClicker, which required an update of the firmware, which I think that utility toasted my iClicker based receiver and it wasn't working. So I've gone back to a little bit older version, and Dr. Zhang loaned me his. So we'll see if it's going to work. Now I've had a number of students calling or coming by my office because they've ordered the iClicker 2, but they don't have it yet. So would you raise your hand if today you have an original iClicker, but you intend to replace it, let's say by Monday? Okay. I can link both of those iClickers together so you get all the credit, but we'll probably have to do it manually. So when you get the new one, register your iClickers on Blackboard. I can't really see whether the registration is working until you have at least one response in the system. So all that really happens when you send in your responses is the receiver is collecting everything by the serial numbers. And so I can manually enter 2 serial numbers if we need to. So just when you get your iClicker 2, put it in on Blackboard. Let's see what the registration looks like. And as long as we get it fixed before the end of the semester, we're good to go. So we'll see how that works. I also made some corrections to the syllabus. So the updated version is on Blackboard. The main thing being I ended up in science by default because I flunked out of secretarial school. And so I had a mistype that only had 30 minutes worth of office hours, which actually was not a bad idea at first. So I tweaked them a little bit. So we'll do Tuesday and Thursday from 12 to 1:30. If you cannot come in at that time and you want to make an appointment, email is the most effective way to do it because then I link it right into my calendar so that I know that I have an appointment. The other is I moved exam 1 to September 16th to avoid the conflict with the Jewish holiday on that Monday. So if you look at the way it's been readjusted, and one of the important things is, at least for the first 3 exams, we're going to finish the material for that exam at least a week before you take the exam. So you're going to have plenty of time to think about things, catch up on 7 chapters of reading, do all those problems, ask me questions and such. So we're going to be doing chapters 6 and 7 before we do exam 1 now, but I moved it around so that the September 14th is really my catch-up and answer question day. So at least for the students who will be out that day, I think the loss of your time in the classroom will be minimal at that point. If you have any questions, again, let me know. All right, so what I want to do very quickly, we're not going to go over the results. I just have this group of questions I like to ask the students because I think this is a good cross-section of biology and non-degree seeking students, and it helps us figure out how to schedule classes, where students are. We're on multiple campuses now. So if you have an iClicker with you, we're just going to go through a number of real quick questions and then also see if the system is working. So it should only take about 30 seconds on each one. You should, I think if you have the original clicker, you get a solid light on vote, it means it came in. Once this window is open, you should be able to put in your answers. In the future, if you want to change an answer, you can press the button as many times as you want. It only records the last response, but it will only accept it once this window is open. Okay. Whatever the default channel is. And I think on the iClicker too, don't you get a feedback? It says submitted. I'm learning about the iClicker too. You get a check mark? Okay. So I mean, right now I've picked up, there's about 100 responses that came in.
Do you know how to change these? Because mine's not on the default channel.
No. I've never had to deal with the frequency before.
You just have to hold down the button.
Okay. So you hold down the power button. Okay, so if you're having trouble getting the response back, the check mark, whatever it is, hold down the power button and then press AA, and that should get you back on frequency. All right. Is there anybody trying to send in responses right now? Okay. We got 234, so that's like the class. Okay. Seems to be working. We're going to find out. Next one. Let's see. Come on. I'm relearning too. I haven't played with. There we go. So if you take in biochemistry, that's going to be it. Okay. We're at 234. Must be it. As I said, I'm just trying to go through these very quickly so we can use this data. All right. What I want to know here is if you are a traditional MD pre-med. Just if you're looking to go to an allopathic MD school like Miami or Gainesville. Okay. Now this one is just any health-related area. MD, vet school, DO, PharmD, PA programs, any health career-related one where you want to deal with sick people or animals. Okay, that's it. All right. So which of these is most appropriate? As a biology major, you're either interested in micromolecular biotech related stuff, marine biology, more ecology, conservation oriented, pre-med, or you're not a biology major. Okay. Interesting one on this one. Very consistently over the years, about 80% of this class has been in the health career field. From what you're showing me here, it's only about 62%. Not sure what that means. I can make some guesses. But. Okay. This is to get a feel for how many are going after the physician's assistant. So you're either a biology major for that purpose, you're a non-degree seeking student doing that, or you're not pursuing the physician assistance programs. Okay. While we're doing that, anybody here interested in a health career field and you're really good at physics? No one. Okay. I didn't mention it. I forgot to mention it last time. There's a second professional science master's program. It's over in the physics department. Dr. Leventuri runs it. It's in medical physics. And so this is a program, what might get your interest, and you might want to learn more about it, is to my knowledge, every hospital has these people on staff, so you'd have the flexibility of going virtually anywhere in the country you wanted to go. The last time I looked into this, starting salaries were about $120K, mid-career was about $180K, and you're looking at basically a 2-year master's level training program. So if that is all interesting at all to you, then look at the physics department, and you might want to talk to Dr. Leventuri. Okay. This one is we kind of want to know a little bit about your background. So are you a transfer student? Or have you taken all your courses here at FAU? Did you come in with an AA degree, or did you come in with less than 45 credits? Are you a non-degree seeking student, or are you working on a second undergraduate degree? Okay. All right. We also want to know how much you work. When I talk to students and they're coming in and crying in my office, you get the sense that everybody's working too many hours. But why don't you just actually tell us? All right. And this isn't for the purposes of stalking. We just want to know which most of the time students who are taking this course live closest to the Boca campus. Okay. So it looks like two people don't really know. Okay. I had to think about it. Okay. And how far away from the Boca campus are you? Do you live on campus, or sometimes? Where's that one or two students who are always driving down from Harbor Branch? Anybody doing that commute this time? Guess I'm not as popular as I used to be. Okay. All right. As you know, we're building the program on the Jupiter campus because of Max Planck and Scripps and Life Science Initiative. It's really where the cluster of the neurobiologists are developing. And so the question is, would you be interested in taking courses up on the Jupiter campus? Just as I know many of you take courses down at Davie. I guess that's as far as okay. Okay. Let's change it a little bit. Do you change your mind or have the same idea if there was a reliable shuttle that could give you transportation from Boca up to Jupiter and back? Okay. All right. Thank you for that information. We're done for today. Okay. So now we're going to start the science. So this is the point where maybe by the end of today, a few of you may wish that I was teaching all of your classes, and another group of you will be quite sure that Dante missed one of those circles of hell in his epic poem, The Inferno. Okay. So here's what we want to do. With this chapter, we're going to do mitosis and meiosis. You've seen this before. At least you know the words. You've seen telophase, metaphase, anaphase. Hopefully what I'll be able to do is show you, give you a little more context to this and a little bit more of the mechanics of what's going on. So the overarching idea here is to understand how the chromosomes behave in meiosis, and then next time when we talk about Mendelian genetics, all of the observations and the very important genetic rules that Mendel laid down about 150 years ago actually make perfect sense when you understand how these chromosomes, basically the chromosome mechanics. So the real emphasis is going to be on how those chromosomes segregate. So the overall, sort of the organization of the discussion will be one, a very broad introduction to a comparing and a contrasting of prokaryotic and eukaryotic DNA organization. I'm not going to go into it in a great detail because especially for the eukaryotic chromatin structure, we're going to look at that in much more detail in a later lecture. But really to set down a very fundamental idea, and that is that structure dictates function. And so we're going to do a lot of comparing and contrasting as we go through the course, comparing prokaryotic organisms to eukaryotic organisms. And there's going to be a lot of similarities, and that gives you that thread of evolution that you can see going through the system. But there's also major structural differences. You already should know this, that eukaryotic cells have these membrane-bound organelles, including the nucleus. And so this change in structure dictates that we have to have a change in the way that it functions. All right. So the first thing that we're attempting to do now, in terms of this iClicker and extra credit, is we're going to start out with one question from the previous lecture and one question from what would be now Chapter 2. Yeah, go ahead and keep your clickers out. There's questions peppered throughout here. These first two are going to be worth 10 points. So again, the participation is 20 points, but you have to answer all the questions. Come in late, leave early, you're probably going to lose that. The second these are to prompt you to be keeping up, reviewing, and looking ahead. And so, all right, we're going to make these a little more high-value questions, and then the rest of the questions that go throughout are going to be worth 2 points. Okay? So the first is, so in the central dogma of biology that Francis Crick put forth many years ago, what is the most important tenet of that central dogma? And I'll go to 1 minute on this one. Okay. So what's the answer? Okay. This next one is also to see if we can actually, since this is my first time with the iClicker 2, let's see what happens when I make this numeric. Can you answer this now with? No, because I got ahead of myself. I was thinking of another question. All right, never mind. We'll stop. Hang on. Let's do this again. Hang on. We're cool. We can do this. Let's try alphanumeric. That'll be okay. Does that work? Does that change?
What do we do?
Well, I don't know. That's what you tell me. Okay, so if you got an iClicker 2, there's directions on the back. It says press the refresh button on your iClicker. Is that right? It's a learning process. That's what science is all about. It's always a learning process. Yes. I'm sorry, what happened? You didn't say something. Yeah, that's what. Now you have to enter. So this one you have to enter text. Well, that's what I wanted to learn how I want to see how all this works. I got these salespeople that keep coming in my office telling me to use this stuff. Yes. Yeah, I think they said they're going to come in. They're supposed to be coming in very shortly. So how do you submit it once you type it in? It probably says press here. Yeah, no, that reading's a bitch, isn't it? It's so useful though. Okay. I'm not going in. We're just. Are we getting? Are we? Is this one of these things you got to scroll down through them and? Was this like texting on an old flip phone? See, I just made this assumption that Apple made it and therefore it would work. It'd have voice recognition and everybody be talking. We may maybe we'll do the numerical ones, but this sounds like this is way too much effort. Or I'll have to play with it a little bit.
Yeah, I think pick short words.
Pick short words. I don't know. It says. Everybody got it? Yes. No. Well, tell you what. We're gonna look. I'll give. The nice thing is I can fudge around the coding. I'll give everybody credit on this one because it's a learning process, so that I don't spend the whole day just trying to get you guys to learn how to use a clicker.
Like, how do you submit it? Oh, okay.
All right. So what's the answer? Meiosis. That's where we're going. All right. So I learned a lot too. All right. Now let's get to the science. So as I said, first what we're going to do is talk about the general organization of genetic information in prokaryotic cells versus eukaryotic cells. But we're going to do this very quickly in a very cursory way because we're going to come back to it in more detail in a later chapter on DNA replication. So you should already be familiar with, you know about prokaryotes, basically bacteria, the most common ones that we're going to encounter. In eukaryotic cells are the higher cells beginning with pretty much fungi and protozoa and coming all the way up to humans. Transitioning between the prokaryotes and the eukaryotes are the eubacteria are the true bacteria. John Wu, several decades ago, started to look at sort of the phylogeny of organisms based on using some molecular data by sequencing. He was looking at something called the 5S ribosomal RNA. And in the process, he discovered these very unusual bacteria-like organisms, single-cell organisms, that he thought were actually older than the current day true bacteria, the eubacteria. So he called them Archaeobacter for the ancient bacteria. Now that we understand them a little better and we've studied them in more detail, they're more like a transition between, they're that missing link between the prokaryotes and the eukaryotes. They're very prokaryotic in nature, but for instance, some of their genes have introns, something that we see in eukaryotic cells. So there's a little bit of a misnomer, but it's hung on now for all these years. Okay. The major focus of the genetics in this course is going to be on eukaryotic organisms. That's where the bulk of the work is being done. That's really in terms of human diseases and such, and the genetics, and where we're going to be going to towards the end of this course in terms of personalized medicine and high throughput sequencing and meta-analysis of genomics and such. We're going to focus on eukaryotic organisms. Here's going to be a major distinction. In prokaryotes, you don't have compartmentalization within inside the cell. So the transcription, that is the synthesis of RNA from the DNA, and translation, the synthesis of the proteins using the RNA, all occur within the same compartment. So it's, for instance, generally in prokaryotes, translation is beginning before transcription has even been completed. In eukaryotic organisms, that DNA is sequestered away inside the nucleus and where transcription occurs. But where does translation occur? Out in the cytoplasm. So they're physically separated from each other. So that's right there, that's that structural difference is going to tell you there's going to be major differences in the way genes are expressed in prokaryotic cells versus eukaryotic cells. Okay. That's really what, so this is this idea of structure dictates function. All right. Going to go very quickly here just to give you a big overview of eukaryotic chromatin organization, because we're going to come back to it in more detail in another chapter. In prokaryotic cells, in bacteria, there's only a small amount of protein associated with the DNA. The DNA is not highly organized. It's basically in a clustered little corner of the cell. In a lot of organisms like E. coli, it's physically attached to the inner side of the membrane, but it doesn't have a high level of organization. And it's only got a small amount of protein associated with it. In eukaryotic organisms, the DNA is organized into what? Things called chromosomes. Okay? But what you know of as chromosomes, we've seen the little pictures of karyotypes, that's only during metaphase in either mitosis or meiosis. During most of the cell cycle, this chromatin is very diffuse all throughout the nucleus. But it actually has a very high level of organization to it. Part of it, and we call it chromatin when you can't really tell discrete chromosomes. It's about 50% DNA and about 50% protein. This was one of the reasons that scientists for so long thought that it would seem just as reasonable that the genetic material would be encoded in protein as it would be in DNA, because they knew in eukaryotic cells, this chromatin, these chromosomes, are about 50% protein, 50% DNA. The main problem is it's a packaging problem. Okay, it's in a human cell. What's larger, eukaryotic cells or prokaryotic cells? Eukaryotic cells are larger, but the DNA is all in the nucleus. So let's be very generous. Let's say the nucleus is 10 times the size of E. coli. But a human cell has 1,000 times more DNA than E. coli does. So in the same space, the DNA has to be packaged about 100-fold tighter. Okay? So it's like trying to pack for airlines now, because you got to pay for every single suitcase you take on. So you try to pack it just short of critical mass. Okay? Eukaryotic cells have to do the same thing. They got a massive amount of DNA, and it's really organized, although it doesn't look that way. So this is the general organization. Again, we'll come back to it later on, just to recognize these structures called nucleosomes. So about half of that protein that's associated with chromosomes are these histones. And so these nucleosomes are made up of a core of 8 histone proteins. And then there's H1. And the DNA is wound around the surface. And it gives you this kind of beads on a string structure. So when they looked at chromatin under the electron microscope, they could see this bead and then a string and then another bead. But it's this level of organization. And when we get into this in more detail, you'll see there's about 6 levels of coiling in here. And the whole purpose is to be able to take this massive amount of DNA and put it into a relatively small space. But again, we're going to come back into this more detail later on. Okay. Another big difference in the way the DNA is organized. Well, pretty much, I'll try to maybe use the term bacteria just so we're clearly distinguishing prokaryotes from eukaryotes. In general, and the problem is there's exceptions to almost everything I'm going to tell you. In general, bacteria have a single chromosome. and it's a circular molecule. So for example, the best studied model, E. coli, has a single circular DNA molecule of a little over 3 million base pairs. All right. Now, all living cells, whether bacterial or eukaryotic, have double-stranded DNA as their genetic material. But in the bacteria, it tends to be one chromosome and it's circular. In eukaryotic organisms, do humans have one chromosome? Anybody here with one chromosome? We have how many? 40 something, right? Let's try 46. Okay, 22 autosomes, one pair of sex chromosomes. How many DNA molecules in each of those chromosomes? In each chromosome? One. For a long time, a lot of investigators thought the DNA was broken into pieces and there might be tens of thousands of pieces of DNA and the protein was holding them all together. It turned out to be an experimental artifact because DNA is so fragile that just handling the cells and handling the material prior to examination broke the molecules. So it's now clear that even the largest chromosome, like chromosome 1, which is millions and millions of base pairs in length, is a single linear double-stranded DNA. Kind of foreshadowing to where we're going in DNA repair later on, there will be a biochemical problem with replicating the ends of a linear DNA molecule, and the bacteria get around this problem by having a circular molecule. We're not going to go into the details now, but again, it's the structure dictates function. Replication of a linear molecule has some different constraints on it that you don't have with a circular molecule. Okay. So the main thing we're going to be looking at here in both mitosis and meiosis is a mechanism for replicating that DNA precisely and then distributing it to the new cells. All right. So let's try this question, since it doesn't require that you type anything in other than one letter. All right. So genetically speaking, prokaryotes differ from eukaryotes because Can you read these from the back of the room? Okay, if you can't, you probably need glasses. No, I just, when I was taking organic chemistry in a big lecture hall like this, I couldn't read the board, but I thought, nobody can see that far. But then I got some glasses, realized, yeah, you actually can. All right. So what's the answer? Okay. So you're also going to have the answers to these iClicker questions. They'll be part of the video cast, podcast, whatever you want to call it. Okay. As I mentioned last time, viruses, important for many, especially for health reasons. As you know, they are not living organisms. They are literally just a piece of nucleic acid wrapped up in proteins. Some of them, especially viruses that infect eukaryotic cells, have some lipids associated with them. In terms of their genetic material, unlike the constraints where living cells, as we know them, have double-stranded DNA as their genetic material, viruses are very flexible. There are RNA viruses, there are DNA viruses, there are circular DNA, circular RNA, single-stranded, double-stranded. There are even segmented RNA viruses, the reoviruses, which are almost like little chromosomes. They're very flexible this way. However, if I gave you a junior scientist question and some new entity, some cell, wasn't sure if it was a cell or a virus, and you started doing some analysis on it, what nobody has found yet is a virus that contains both DNA and RNA. They are one or the other, as opposed to living cells, as we know them, will have both DNA and RNA. Okay. So if we want to look at cell replication, we really have a few critical steps we have to look at. One is this has to be an extremely accurate process. What would be the consequence if DNA replication was only kind of accurate? This is genetic information being permanently transmitted to the next generation. Any mistakes made during replication are the equivalent of a mutation. So this has to be a very precise process. And when you look at it experimentally, it is. These replication systems have a high enough level of fidelity, of accuracy, that the error rate is only on the range of about 1 in a billion nucleotides. So each of our cells contains around 4 billion nucleotides, so each time our cells replicate, there's probably on average 3 or 4 bases out of those 4 billion are wrong. Now, there are no enzyme systems out there that have that level of accuracy. But as we'll get into later on, there are some fail-safe checkpoints, DNA checkpoints, and by doing this through 3 different cycles, you can reach this level of fidelity. For now, just understand that DNA replication is extremely accurate. And in eukaryotic cells, this whole genome has to be replicated and then distributed to a specific place in the new daughter cells. So we'll have to talk about that process. And then you actually have to separate or divide the cells. Okay. In bacteria, this seems like a fairly straightforward process. So under optimal conditions, at least the literature tells us E. coli can replicate in about 20 to 30 minutes. In our hands, it's more like about 45 minutes. But let's go with this 20 or 30 minutes. This is a very fast process. If you commit the entire 30 minutes to replicating 3 million bases of DNA, and later on we'll talk about how it's bidirectional, so you really only have to do about 1.5 million bases. But if you do the math, these cells have to be synthesizing DNA at a rate of at least 3,000 bases per second in order to get the job done. And only being able to make a mistake once out of every billion nucleotides. Okay. If another little calculation you can do, you should already know that DNA replication is semi-conservative, so the DNA strands have to unwind during DNA replication. If you do the calculations here, you also realize that that unwinding has to be moving at about 3,000 RPMs. So this DNA strands are spinning at about the same speed as the engine in your car if you're in the slow lane on 95 doing about 80. Okay. So if you have a bit of a gearhead, that's why I understand these things. But, all right, so just doing this in a diagrammatic form. So here is the DNA. So the prokaryotic cell, again, has a single circular molecule. It's supercoiled. We'll deal with that later on. There is an origin of replication. That's simply the point where replication begins. Okay, the molecules replicate, and they get segregated off to the 2 different sides of the cell. And then you have cell division. It's a pretty straightforward process because you don't have this compartmentalization, and there's only one DNA molecule to deal with. Okay. Another molecule, another common DNA, type of DNA that you see in bacteria, but they're very rare in eukaryotic cells, are plasmids. You can think about them as little tiny mini chromosomes. So they're circular molecules. They have an origin of replication. They have to be replicated. They have to do everything this chromosome did, except they're just much smaller. And one example is one called the F factor. We're going to find out later on that E. coli actually has a sex life. Hopefully it's not more interesting than yours. It's a process called conjugation. but there's actually a mechanism for genetic exchange between two cells. The genes needed for this process are on this plasmid called the F factor. And it's a fairly large plasmid, but even there, it's about 1% the size of the main chromosome. Anybody know what else, why evolution is selected for plasmids? I'm sorry, what? I've been married so long, I'm almost deaf. You got to speak up.
It was theory.
It was theory?
Theory. You know, like an iPhone.
Oh, Siri. Oh. See, I know what that is. I have an iPhone. I just didn't hear it. Okay, the real answer is molecular biologists needed vectors for cloning purposes. And we use, and plasmids are very common. So a lot of the molecular cloning techniques, if you decide to take the biotech lab later on, we do some of this cloning. These plasmids have been highly manipulated now. They have no resemblance to their natural sources, but they started from naturally occurring plasmids that have just been, their sequences have been manipulated over the years. The main thing that the bacteria do with this is where many of the drug resistance genes reside. So this is a major reason that drug resistance can move through a bacterial population very quickly. There are mechanisms for quickly transferring this genetic information, sometimes even across species barriers, something called horizontal gene transfer. And this is one of the main, and so if you're in a place, especially with high uses of antibiotics, you select for this. So this is one of the main reasons why the drug resistance bacteria and such are such a problem at this point. So these plasmids, they're common in bacteria. We'll talk about them later on. They're going to be used as a major mechanism for letting us map genes. Before we were able to sequence entire genomes in about a day. And they're also used for molecular cloning purposes. Uh-oh. I'll just give you that one. It'd take you the rest of the afternoon to spell out prokaryotes, so. All right. Now it gets a little more complicated. You got eukaryotic cell replication. This is really what we're going to focus on for the rest of the talk. We've got multiple chromosomes. We have very large molecules. We have these molecules and these structures. These protein DNA structures are inside a nucleus. That nucleus is going to have to be dismantled and has to be rebuilt. So a much more complicated process. Let's just kind of walk through this. So the DNA is distributed among multiple chromosomes, in contrast to being just one that you find in bacteria. And they're separated from the cytoplasm by this membrane structure, the nuclear envelope. And while it may look disorganized, let's say if you're looking at chromatin in the microscope, there actually is a very high level of organization in that nucleus. And there's a system of cables, the nuclear matrix, basically protein cables that run through that whole system that help organize and keep certain parts of this chromatin in certain places. So it's a very highly ordered structure that's now going to have to be broken down and then built back again. Okay. Now, different species, each species will have a characteristic number of chromosomes. What you can't predict is how many chromosomes will be present based on the, let's say, developmental complexity of the organism. Drosophila has 4 sets of chromosomes, including the sex chromosomes. Humans, we have 46. But there are other organisms, other mammals, they have just different numbers. So the take-home message is you can't predict how many chromosomes are going to be present in any given species. Okay. Now, the first thing we're going to talk about is mitosis. So there's 2 major types of cell division, mitosis and meiosis. If we're not dealing with, I'll use a term that a lot of students haven't heard before, germline cells. So the germline cells refer to the egg, the sperm, any of the cells that are destined to be used for reproduction. If we're not talking about germline cells, these cells will replicate by mitosis. So these are diploid cells, meaning that there's two sets of chromosomes, homologous chromosomes. You should be familiar with this. All right. So anybody know what this is called? These are karyotype. But these are metaphase chromosomes. You make them by treating cells with drugs that block them at metaphase so that the mitotic spindle can't actually function. And you can see the different shapes. So in the human karyotype, the chromosomes were originally numbered based on their size. The number 1 was the largest all the way down to 22. What do you know about 21? What's special about 21? For Down syndrome, when it's a triploid, we'll talk about later on how these aneuploids aren't tolerated very well in mammals, in animals at all. But small ones, okay. Well, we now know 21 is actually the smallest chromosome now that it's been sequenced. All right. And here's the 2 sex chromosomes. So if this was a human cell, this would be male or female? Male? Why did you say male?
Well, it looks like that's a smaller chromosome like this.
No, the Y is much, much, much smaller. This would be female. We will talk about a couple of genetic systems. If you didn't know if this was some new species, I'd write these junior scientist questions. This was some new species. This actually could be the male of the species if it's in what is called the ZW system. But we'll talk about that later on. So what you really have is homologous chromosomes are 2 nearly identical chromosomes. Could we do genetics if the chromosomes were identical? No, we got to be able to see some differences to them. So here is the A locus. So the term locus is used as the address where that gene resides on the chromosome. So this is the A locus, and here's the corresponding A locus here. But we have alleles. All alleles mean are different versions of the same gene. Okay? So how many different alleles can you have in a diploid organism? What's the maximum number? The maximum is 2 alleles. What's the maximum number of alleles that can exist for any given gene? Is it only 2? Well, technically, you change a single nucleotide, you've created a new allele. So it's quite common that out in the wild type, you have the wild type, which is simply the allele that occurs most often out in the population. But you might have 20 or 30 different alleles of that particular gene, but any given diploid, any individual can only have a maximum of 2 alleles. All right. We're going to skip that one for obvious questions too, for obvious reasons. Okay. So now let's look at the structure in a little more detail. The circular chromosomes found in bacteria, basically an origin of replication. That was pretty much good enough. Things are going to be a little more complicated with eukaryotic chromosomes. All right. So first of all, each chromosome, as I said before, is a single linear double-stranded DNA molecule. Although there's this extensive level of coiling that we'll talk about in another chapter, that we really can only call them chromosomes when we see them at metaphase, as I showed you in the previous figure. But during most of the cell cycle, which is interphase, these chromosomes are very diffuse. A lot of the coiling has been reduced, relaxed, and it just looks like if you stain for DNA and you look through the light microscope, the whole nucleus is just sort of uniformly covered with this chromatin. All right. So just before cell division begins, one of the first things that has to happen is this chromatin is going to begin condensing. You're going to begin getting your clothes together, getting them into the suitcases, getting things packaged up. What's having to happen here is we need a mechanism to package up all this DNA and be able to transport it to opposite sides of the cell during cell division. There's 3 critical parts to this chromosome, and we're going to come back to this stuff in more detail. And when I get done telling you about this in more detail later on, I'll tell you how you can go home and make your own chromosomes. Okay. 3 key parts. You have to have these. You have these 3 main parts. Then you can build the artificial chromosome and win the Nobel Prize. The centromere, the telomere, and the origin of replication. So let's just go through each of those. Okay. Oh. All right. Listen to me. This gets confusing. Okay. The centromere is the point on the chromosome where the microtubules attach so that they can pull the chromosomes to the opposite poles of the cells. The centromere is a DNA sequence. The attachment of that microtubule at that point and all the proteins that are associated with it to bond that cable to the chromosome, that forms the kinetochore. So the kinetochore is the structure that forms on the centromere when the microtubules attach. The centromere is a DNA sequence. It tells those proteins, those kinetochore proteins, where to bind to the DNA and to the microtubules to bond and give you a structural joint there. Okay? So the centromere is the DNA sequence. The kinetochore is the actual structure that forms once the microtubules are in place and all of the associated proteins. All right. To show the essential nature of the centromeres were first isolated from yeast cells. Are yeast prokaryotic or eukaryotic? They're eukaryotic, but they're small, easy to work with. And so they were the first eukaryotic organisms that had good genetic manipulation techniques, molecular techniques. In John Carbon's lab out at UC Santa Barbara, I went out there one time. I visited some people, and I don't know how you get any science done there. I don't know if you've been to Santa Barbara, but the campus is sort of sticks out into the ocean, and it's right on the beach, and this window looks like some postcard. And talk to the grad students. They go, yeah, we get a gel started, then we go down to the beach, that sort of thing. Great place to go to grad school. What his lab showed was if they removed the centromere from a chromosome, it was lost during cell replication. So it provided the scientific evidence that the centromere is absolutely essential for the segregation of these chromosomes. So some terms you've probably seen before: metacentric, submetacentric, acrocentric, telocentric, if I get this thing out of the way. Okay. These are relative terms, not absolute terms. They've been used over the years to help describe chromosomes, especially in these types of karyotyping. All right. So there isn't a nice fine line between here it's submetacentric and now it's telocentric or something, or it's metacentric. They're just relative terms. So if it's approximately in the middle, we'll call it metacentric. If it's clearly offset to one side or the other, submetacentric. Getting close to the end of the chromosome, acrocentric, or telocentric, like the X chromosome in Drosophila, then the centromere is right at the very end of the chromosome. Yes? No, it's just where it happens to be, as long as it's there. Is it going to matter if a cell has 2 centromeres? Is that going to be a problem? And breaks the chromosome. So later on, we'll talk about some gene rearrangements or DNA chromosome rearrangements that actually lead to dicentric chromosomes, and they're extremely unstable. The microtubules attached to both, they're pulling in opposite directions, they end up breaking the chromosome. Okay, let's try this one. Okay, so which structural component is the attachment point? Where's microtubules? So this is that little gray area where I know my tricky question. Do I write tricky questions? No, but they're detailed. All right. So I've told a lot of students over the years, when you're staring down at me and you're about to do my quadruple bypass surgery, I want you to know everything. I don't want you to generally know what you're doing. Good enough to pass the exam. I want you to know everything. So science is the devil's in the detail. That's for science. Okay, looks like I'll go to 1 minute. Okay. All right. About half of you said centromere, half of you threw the coin in the air. All right. So the component is the attachment point where the microtubule goes to. The kinetochore is what forms at that attachment point. Okay. Sometimes the detail is very fine, but it's there. Okay. So the centromere is the DNA sequence. If you're isolating the centromere from some new organism, you'd be looking at a DNA sequence, not a protein. Okay. The third component is the telomeres. I mentioned earlier that later we're going to have to talk about a problem that the cell has to face in replicating a linear double-stranded DNA molecule. And when this was recognized, and once it was realized that linear molecules are very unstable in cells. Later on we'll be talking about chromosome rearrangements and such. In the early days of Drosophila genetics, there were some geneticists, they called them chromosome mechanics. They basically take the flies, put them in an X-ray machine, bombard them with X-rays, and the X-rays, the high-energy X-rays, literally just slice through the DNA and make double-stranded DNA breaks. Those broken chromosomes are very unstable, and the cell just starts knitting them back together. It doesn't want those double-stranded breaks like that. So that told us that the telomeres, there must be something special about those telomeres. And there is a very special structure to it. We'll get into that later on. The only thing to kind of talk about that, just a couple of things that lead up to it, is these telomeres are very dynamic. And there's an enzyme called the telomerase, which is really responsible for maintaining these telomeres. And so what you find is this telomerase is very active in young cells and then goes quiet as the cells reach homeostasis, kind of a quiescent stage. The telomeres begin to shorten due to nuclease action and such. And that when they reach a certain point, it becomes a trigger for apoptosis or programmed cell death. A couple of things that were learned out of this. One, everybody know about Dolly the sheep, cloning Dolly and that sort of thing. But they didn't realize, well, once she reached, I think, 5 or 6 years old, she started developing arthritis and some sort of old age conditions, and eventually they had to euthanize her. When they looked at the telomeres, what they realized is she was basically born, it was under this manipulated system, but she was born with old telomeres. All right. So we know that has to do with aging. The second is, in virtually every cancer cell that's been looked at, you can find that the telomerase has been reactivated. So there's a lot of research into what's happening in this transition from a normal cell to a cancer cell, which is really an uncontrolled cell proliferation that seems to involve this telomerase. But for now, we'll just say you have to have this special structure on the end, the telomere. So we have the centromere, telomeres, and an origin of replication. All right. So we'll give you another one that should be pretty. All right. So this is just sort of an introduction into junior scientist questions. So you got some new population of cells you're looking at. Which of these would you find would be most likely true? A pretty good class. Usually in a class like this, with this kind of question, there's at least 2 or 3 answer E's. Okay. All right, looks like we're there. All right, so what's the answer? Yeah. Okay. Yes. Okay. Okay, if you put in multiple answers, it records the last one you entered. All right, so we've talked about centromere, we talked about telomeres, now we have the origins of replication. The molecules in bacteria are small enough, there's only one origin of replication. Later on, we'll talk about how in eukaryotic chromosomes, these molecules are so large that if there weren't multiple origins of replication, in some cases, such as Drosophila, it would take nearly the lifetime of the animal to be able to replicate the cells once. So there's multiple origins of replication. We won't talk about that later. We'll talk about that later. Right now I want to talk about a confusing point. It's a terminology question. So here we have one chromosome. We have the centromere, here are the telomeres, and it turns out there's origins of replication all up and down the chromosome. We won't worry about that for now. When we go through DNA replication, so the first step is going to be we have to replicate the DNA and make 2 chromosomes so that now we can send 1 chromosome to each of the 2 new cells. During that replication, these 2 chromosomes, the original and the newly replicated, are held together at the centromere. So they're really one entity at that point. Here's the confusing part. Prior to replication, we call this one chromosome. We also have used the term as one chromosome for these replicated, though there's really 2 chromosomes there. All right. But until they split at anaphase, we'll still call them one chromosome, and then all of a sudden we're going to have 2 chromosomes again. Yes?
Do they call them like one chromosome because they're like
Yeah, they're duplicates of each other. So these are, to make it even more confusing, each of these is a chromatid. So these are sister chromatids, telling you these 2 molecules should be genetically identical. So these are 2 identical chromosomes that are the result of DNA replication, and they're held together, so that they're carried together through mitosis and meiosis. They're held together at the centromere. Okay. So to be able to do this, and one of the other differences between bacterial DNA replication and eukaryotic systems is in eukaryotes, there's a particular phase in the cell cycle where the DNA is replicated, and it's completely distinct from when mitosis and meiosis occurs. So you should be somewhat familiar with the cell cycle. We're not going to go into it in a lot of detail, but just enough as to what's important for what we're talking about. Now, as we go through this cell cycle, it's important to point out there are checkpoints along the way. So there are check mechanisms that are checking to make sure the DNA is not damaged and it is really ready to be replicated. Once the cells enter this S phase, once they enter a replication, they're committed. They have to complete it or it's lethal. So the idea is, are the cells ready to enter into this S phase, or do we have to stop and make some repairs? Some of the oncogenes, some of the genes that are involved in cancer development, one we'll talk about later on is the one responsible for retinoblastoma, RGB. It functions in this checkpoint. When these systems aren't working, then this leads to mutations and uncontrolled cell proliferation in a cancer situation. Okay. Interphase in a eukaryotic cell cycle is all of the time except M phase. So if the cells aren't physically going through mitosis or meiosis, they're in interphase. All right. So you have the G1. We're going to see this. You got G1, S, and G2. Those three together make up interphase. Unlike replication in bacteria, replication in eukaryotic cells is slower. It's more complicated. The rate of replication is slower. There's a lot more DNA to replicate. And so going through this whole cell cycle and getting ready for the next round of replication can actually last several hours. This checkpoint I was talking about where RGB functions, this G1 to S transition, which I'll show you in a figure in a minute, this is critical because this is the point of no return. If the cells enter into the S phase, they must complete the cycle or the cells are going to die. Where am I going here? Come on. There. Did I throw it in twice? Oh, I guess I put it in twice. Almost. So you're going to start, you got G1. The cells are growing, they're getting prepared. This is the critical one that we're going to look at, S phase. This is the period in the cell cycle when the DNA replicates. So it gets its name from synthesis, from DNA synthesis. This is, oh, that's why I just put this figure back. So this is when you went from one chromosome to these two chromatids. So these are genetically identical chromosomes called sister chromatids. They are the result of DNA replication. All right. This is also when the cell is having to make a lot of histones because it has to repackage all of these chromosomes into chromatin prior to going into the next phase, which is G2. So G2 is that period of time when the cells come out of the S phase. So now they have a fully replicated set of chromosomes, and they're progressing to M phase, which is the part of the cell cycle where the actual chromosome segregation is going to occur. All right. So here's the sort of traditional cell cycle. So we can start. We have two individuals. So we have our two new cells. We can just start there. We go through G1. When it hits this checkpoint, if there's some DNA damage, let's say the cells have been irradiated or exposed to chemicals, something has damaged the DNA, these proteins like RGB that function here put the cells out into so-called G0, which takes them out of the cell cycle until everything can be repaired. Then they can re-enter the cell cycle. Then you have G1. This period down here, S, this is when the DNA is duplicated. So at G2, the cells have twice the DNA content. How many? But do they have the same number of chromosomes? Technically, yes. Do they have the same number of DNA molecules? No, they have twice as many. All right. Then there's the G2M checkpoint, and M is either mitosis or meiosis. And again, everything G1, S, and G2, that whole period is interphase. Any question?
Can you repeat what you just said? The same number of chromosomes, but double.
Okay. So let's say this is a human cell. At G1, we have 46 chromosomes, right? And how many DNA molecules? One DNA molecule per chromosome, so 46. We go through S, we come out over here, we still say that we have 46 chromosomes, but they're sister chromatids held together. So there's actually 92 DNA molecules. And then when they split during anaphase, then we're going to have twice as many chromosomes. So it's just a technical point of what the terms that we use. Okay. So here are the 6 stages of mitosis. Again, you've seen these before. What I'm really going to be doing is focusing on how the chromosomes behave, how they segregate, and what's different between mitosis and meiosis, and how that changes the outcome of the final process. All right. So we have prophase. Prophase is just that beginning point. The chromatin is going to begin to condense. You can't really see chromosomes yet, but that condensing part is starting. Maybe easier to see this in these really nice figures. So these figures up here are what is called FISH, for fluorescence in situ hybridization. So in situ means in place. And so, and the fluorescence is because they've used the fluorescence tag. So what's happening here is, and here's where I get in trouble because I'm colorblind, but this should be blue. This is probably a dye called DAPI. It's just a dye that binds DNA. So whenever you see blue, that's where the DNA is going to be. This fluorescing yellow, orange, whatever this has been labeled with an antibody against actin. So these are the microtubules that make up the cytoskeleton. So what you're seeing is the cytoskeleton in the cell, and here is the nucleus. This is interphase. So you can see here that the staining is getting a little darker. The chromatin is starting to condense now, and so there's a little higher concentration of DNA. You get a little darker staining. The centrosomes, we're not going to do this in a lot of detail, but this is basically the anchor point on the outside of the cell. So the microtubules have to attach to the outside of the cell and to the chromosome so they can physically provide the traction forces needed to pull the chromosome from the center of the cells to the outside of the cells. Okay, so the prophase is just the very, very early stages of mitosis. Prometaphase, we're just moving things along a little bit more. Most importantly here, the nuclear membrane disintegrates. Remember, these chromosomes have to be out free within the cell. The microtubules are starting to come into that nuclear region. They're trying to find the centromeres and start attaching to these condensing chromosomes. The microtubules attach to the kinetochore of one of the sister chromatids. Here's going to be the important point. All right, and if you haven't figured it out yet, on slides when I use really garish colors or they're bold, or I always use the term important point, key point, that translates to exam question. Okay, here's the most important point, I think, in this discussion. In mitosis, those 2 sister chromatids are held together at the centromere. They're going to split. The centromere is going to break. In meiosis, in the first round, it's not going to break. And that changes the behavior of the chromosomes. So now what you have is microtubules attaching to the centromeres on each of the sister chromatids, because it's literally going to rip the chromosomes apart as it pulls the chromosomes to opposite sides of the cell. All right. So that's what you're seeing forming here and down here in cartoon form. So you're anchored at one end out here on the edges of the cells, and the other ends of these cables, these protein cables, are attached to the centromeres. All right. Here's the main distinction between mitosis and meiosis. It happens at metaphase. So metaphase is the chromosomes are now completely condensed. You can recognize them in these karyotypes. As I said before, it's at this point in the cell cycle when you can add drugs that will cause the microtubules to dissociate, and you freeze the cells in metaphase, and then you can take pictures or do whatever type of analysis you want to do. In mitosis, the chromosomes are going to line up in a single line. And when I get to meiosis, I'll compare and show you what I mean. So they're lining up along this imaginary center line called the metaphase plate. All right. In this diagram, how many chromosomes are in this in a diploid cell from this organism? How many chromosomes do we have? 4. How many pairs of homologous chromosomes? 2. The blue ones will have to be arbitrary, we don't know. But the blue could be the maternal chromosome, and the red would be the paternal chromosome. In metaphase, it doesn't matter what the order is. All the chromosomes are lining up through the center of the cell. There's microtubules attaching to the centromeres of both sister chromatids because the centromere is going to break when we go to the next phase in anaphase, and that's what you see up here with chromosomes. So now we went from, so we had 4 chromosomes. So we still have 4 chromosomes. And now, how many chromosomes do we have? 8. But we had 8 DNA molecules here, and we still have 8 DNA molecules. It's just our convention of saying that this is one chromosome while they're still held together. All right? So these are the daughter chromosomes. They're being pulled to opposite sides of the cell. And this is with telophase, the chromosomes reach. So metaphase is when the chromosomes are lined up. Anaphase is the actual traveling. There aren't just, artificially we put the discrete points in there. This is a continuous process. Telophase, the chromosomes have now reached the poles of the cell, and then you go through cytokinesis, where you have the cytoplasm divides, and you get 2 cells. So that's what you see here. The cells divide, and now you have 2 genetically identical cells. And this is the process of mitosis. This happens in somatic cells. Somatic cells are any cells that aren't going to give rise to the germline. Okay. Just to touch on this a little bit, to show you how this traction, you got to think about this kind of in an engineering physics sense. There's got to be some forces that are going to pull the chromosomes from the center of the cell, break that centromere, and bring it out to the side. So these microtubules are made up of a molecule called tubulin, which is a structural protein, and there's other proteins involved in it called that function as molecular motors. But basically, these microtubules shorten. They begin to dissociate into tubulin subunits, and it's the shortening of these microtubules that are actually pulling the chromosomes and providing the force. When I was a grad student, one of the labs actually was studying this process and actually had a laser system that could go in and individually break microtubules with the laser system and then see what happens to the chromosomes. Okay. So before we jump into meiosis, we'll give you a break to look at this question. All right. And the answer is, yeah, most of you got that. There are a few lost souls out there, but it always happens. Okay. So this is just another way of looking at these changes in comparing these chromosome numbers with DNA molecules. So here we are in G1, 4 chromosomes per cell and 4 DNA molecules. This is what we usually refer to as a 2N cell. So N is the number of sets of full sets of chromosomes. So in a diploid organism, N will always be 2. In most, I don't know if there's any exceptions. If there's any exceptions in animals, I don't know about them. In animals, they're diploid. Plants do what they want. Plants are polyploid, and we'll talk about a few cases later on where they can be hexaploid, 6. A lot, I think, bananas are triploid. That's why they're sterile, these sort of things. But for our purposes, we're looking at diploid cells. So this is 2N. We go through S phase. We still have 4 chromosomes as we go into G2 because the 2 sister chromatids being held together, we're calling it 1 chromosome, but it actually has 8 DNA molecules. Then we go through prophase, and all of that stays the same until we get to anaphase. And now magically we have 8 chromosomes, only because the 2 sister chromatids are now separated. But if you follow the DNA molecules, you still have 8. And each cell at the end has 4. So the math has to add up. You start with a cell with 4 DNA molecules, you end up with 2 cells with 4 DNA molecules. They're genetically identical. So the consequences of mitosis are, here's the purpose: 2 genetically identical cells with a full, complete set of chromosomes. Now, what does happen, and we'll talk about this later on, there isn't the cytoplasm when the cells go through cytokinesis. The cytoplasm is sort of roughly divided in half. But organellar structures like mitochondria, there is no mechanism that partitions exactly half of the mitochondria to each cell. So, and we'll talk about how even the mitochondria within a single cell can be genetically variable. So the organelles within these 2 daughter cells may not be genetically identical. But at the nuclear level with the genome, they should be. All right. What we're really trying to go with this is meiosis. So I'm going to go through, so we've got the main background. What I can focus on is showing you how it's different at metaphase so that the genetic outcome is very different. So meiosis is a specialized form of cell division. It's used exclusively to generate the gametes, the germline cells, eggs, sperm, whatever you want to call them. These are the actual cells that are going to be used in reproduction. So they're going to come together at fertilization to give you another diploid cell. It really provides 2 driving forces in evolution. It gives different genetic combinations by putting the parental and the maternal chromosomes in different combinations, and through the process of genetic recombination, which we'll cover later on. Everybody's done the big A, little a, and they move around. Okay, yeah, well, I'm going to tell you a lot more about it, probably more than you want to know. Okay. The other is meiosis has to precisely divide the chromosomes in half. If you're going to create a 2N individual, you can only have N in each cell. So you got this process has to put in genetic variability and precisely divide the chromosomes in half. All right. So a couple of differences when you compare them. Mitosis, we took you through 1 cell division. Meiosis actually requires 2 cell divisions. Meiosis I is the specialized one we'll talk about and probably get most of the way through it. Meiosis II is mitosis. It's exactly the same. Okay? So mitosis gives you 2 cells with the same number of chromosomes. Meiosis is going to give you 4 cells with half the number of chromosomes. So we started out with a 2N cell, then we went to 4N, and then we ended up with 2 2N cells. Now we're going to start out with a 2N cell, go to 4N, but we're going to end up with 4 1N cells. Okay. The key point here is, just like with mitosis, DNA replication still occurs in S phase. So the G1, S, G2, all of that's the same. The difference is going to be what happens in prophase and at metaphase. All right. Now, this is the only figure in the textbook that I disagree with that I found. I think it's numbered wrong. I think it's Just doesn't follow conventions. This would be a 4N cell because this is going to be the cell. This is going to be 4. Is this going to be from G1 or G2? How do you know it's G2? You have the sister chromatids. Okay, so it's 4N at this point. It's got 4 full sets of chromosomes. Meiosis I is called this reductional division. That makes it two 2N cells. And then the equational division, which is exactly, this goes exactly like mitosis, is now you have 4 1N cells. Okay. Now, in a very important process that we're not going to go to in any detail, but you need to be aware of it. Prophase in meiosis is much more complicated than in mitosis, because this is when genetic recombination occurs. So the homologous chromosomes pair up with each other through a process called synapsis. So what you really have now is going to be a tetrad. You're going to have 4 chromosomes coming together. You have the homologous sister chromatids from the parent, from the father, and then from the mother. You've actually got 4 chromosomes that are lining up from telomere to telomere in exact registers so that they can exchange genetic information. Later on, we'll talk about how if that process isn't exact, you're going to get something called unequal crossing over, and one chromosome is going to gain information, and the other is going to lose it. It's still not clear how those chromosomes find their homologous counterparts and how they align so precisely. But that's what occurs during prophase, and that's when this genetic recombination is going to occur. So they're called bivalents or tetrads because they're so closely associated at this point, at least under the light microscope, you really can't discern them as 2 separate chromosomes. And so this is where this crossing over occurs. They kind of show it in these harlequin kind of chromosomes where now you have some blue DNA on this red chromosome. And similarly here in the chiasma. We'll talk about all this in much more detail later on. Just be aware that at this point in meiosis, this is when genetic recombination occurs. This is when the big A and the little a get started, and the big B and the small b start getting swapped around. Okay. This is the key point. Besides prophase being different and having this formation of the synapsis, which gives you this opportunity for genetic recombination, the difference is how the chromosomes align at metaphase. Here's what we saw over here in mitosis. Chromosomes just lined up one above the other. At metaphase, the 2 homologous chromosomes line up side by side. They're already synapsed with each other. So these chromosomes are all synapsed together. Okay? But this is, if this is chromosome 1, this is chromosome 2, this is all copies of chromosome 1. The 2 copies that came from the father and the 2 copies from the mother, and then we went through S phase. So we got 4 DNA molecules here. Are the 2 red chromosomes identical? Assuming recombination didn't occur. Yeah, they should be. The blue, are they the same as the red? No, because this came from one parent, this came from the other parent. Now the difference is, when these chromosomes separate, the centromere is not going to break. It's going to pull the 2 sister chromatids from one of the homologs, either the maternal or the paternal one. And that's what's different than with mitosis. So as we go through here, they line up. The centromeres don't split. And then at anaphase, you're still pulling the 2 replicated chromosomes. So in this case, the red homolog is coming to the right, and the blue homolog is coming to the left. Now, to preface what we'll see with Mendel, let's say the blue is the maternal chromosome. Just because the blue chromosome 1 lines up on the right, as we've drawn it, has no influence on whether the red or the blue chromosome 2 lines up on the right. Okay? And this now will lead to Mendel's rule about independent assortment. But that's all he was really looking at. Okay. So now they separate. It goes all the way through. We got telophase. Okay. So if we look at this point, this cell, these chromosomes come over here. Is this cell 2N or 1N? It's still 2N. Okay. Would it be, is this, well, if it's 2N, it sounds like mitosis. Is this cell genetically identical to this cell? No. Because this one is carrying 2 copies of one particular homolog, not 1 copy of each homolog. Okay? So it has the same amount of DNA, but they're not genetically the same. Okay? As I said, meiosis II is functionally equivalent to mitosis. So you're going to go through, the centromeres are going to divide. So now those 2 copies of the homologs are going to separate. And let's see. And you're going to get 4 cells that are now what we're going to call haploid. That's where we get the term. So diploid is 2 copies, 2 full chromosome sets. Through meiosis, you get 1 chromosome set. So that was the reductional phase. You reduced the amount of genetic material, the number of chromosomes, by exactly half. Let's see. All right. Let's try this one since it only requires an input of a number. This is going back to sort of the learning curve on this iClicker. So if I make this numeric, see if you can make that work. Well, 2 people have. Are you figuring out? I mean, the main thing, are you able to? Oh, you don't know what the answer is yet. I need to proofread better. It's chromosomes will have how many? The "the" doesn't belong in there. I'll blame it on my grad student. It's that secretarial school thing I flunked out of. All right. So it looks like we can handle numbers. Can you handle, in the future as I write these, can you do texts of, let's say, 5? What's, for the silly ass way you have to do the input of the text, can you do, what should I limit it to, 5 letters? To get done in some reasonable amount of time? So very short.
26 letters.
Really?
Alphabet. It's just up and down.
Oh, good God. We've got to get Apple to redesign these iClickers. I'm sorry, what? Okay. I'm sorry, I only got about half of that. But we'll learn. Maybe it was something insulting, but I just didn't hear it. But you'd be surprised what I can hear. I have selective hearing loss. Beginning to understand why my grandfathers never wore their hearing aids. All right. So we'll give this. Oh. Okay, this only got a couple of slides left, so we'll actually get done. All right. So what's the answer? 10. You feel so good about yourself. Got one right. All right. So this kind of summarizes the importance of this in terms of the genetics. It's this process of meiosis that leads to this genetic diversity. And it's really, just to repeat myself, there's two major mechanisms that are happening here. All right, so to put it more in the context of some of the terminology we're going to use, is first is genetic recombination. What that really does is it puts different alleles of genes into different linkage groups. All right, so later on you'll understand what that means. Also, what we're really focusing on here and how we'll relate this back to Mendel's work that we'll talk about next time is this randomization and distribution of the chromosomes. So that the progeny, the kids, whatever you want to call them, while they're genetically very closely related to their parents, they are in fact genetically unique because they carry different homologs of the different chromosomes, some from the father, some from the mother. So it's this sort of reshuffling of the genetic deck and the genetic crossing over that puts these genes in different combinations with each other, different alleles, that really becomes two of the major forces that drive forward in evolution. And, okay, so as I said, I target 20 minutes till, and this is the wrap-up question for today, so we made it. Now on Wednesdays, if you have questions when you come down here, I may not be able to answer them because I really have to hustle off to the seminar. So I'll remind you as you're answering this question. What did I do? Oh, more and more reasons not to do this. All right. I think I may end up with buyer's remorse before I'm done here. Okay, while you're doing this, if you have any concerns about your future, maybe you do, I don't know. You're welcome to, and how many of you in here are actually enrolled in the seminar? I know a number, okay. So I'll be walking over to engineering. Dr. Fairbanks will be there. He's going to be talking about some career options. Basically, if you're not really interested in health-related areas where you're actually dealing with patients directly, most of his experiences come from pharmaceutical companies, and you don't see yourself as a bench scientist doing experiments, but you like the business, you like the industry, it's a good working environment. He's going to talk about a lot of other job opportunities that you can do with biology and some other training and what kind of training you need. So you may find it interesting. It may be the most important lecture you hear this term. Just depends on how you see your future. Otherwise, we're done for today, and I'll enjoy the weekend, and we'll see you on Monday.